Hey there everyone, welcome to part 3 of the discussion of your pharmacodynamics. So we'll be talking about your types of your drug receptors. I want to correct things class. In previous videos, I've mentioned about the mechanism of the drug in pharmacokinetics. I've mentioned about the absorption, digestion. It should be distribution, I'm sorry. Absorption, digestion, yes, metabolism, and excretion. Okay, now let's discuss about the types of drug receptors. There are actually there are actually four types of your receptors. We have your ligand gated receptors. Okay, your ligand gated receptors. We have also your enzyme link receptors, your G protein and coupled receptor, should I say, and your intracellular receptor. So there are four types. We have your ligand gated receptor, enzyme link receptor, G protein, intracellular receptors. So a particular receptor has a specific site to bind to molecules or ligand. This is called lock and key model. Okay, that is your lock and key model. Let me discuss about your ligand-gated receptor. What about your ligand-gated receptor class? In ligand receptor, the cell membrane requires your specific ligand gated channels for the movement of hydrophilic molecules to it. There. So you can see class, you can see that the ligand there binds to the receptor. And then the receptor, you see that? The receptor gate opens. An example of that is your acetylcholine. They binds to your nicotinic receptor. So the sodium, as you can see, they enters now the cell. And then when they enter the cell, electronic chemical gradient is now created and then resulting in the depolarization. So the receptor gate opens, sodium enters the cell, there will be now electrochemical gradient that is being created, which results in depolarization. So that is what's happening in ligand gated. How about, sir, in your what we call enzyme link receptor? In enzyme link receptor class, the transmembrane of the receptor are enzymes or closely related to your enzymes. An example there, okay? An example of which, as you can see, class, is your tyrosine receptor. The binding of the ligand causes now the conformation, which results in aggregation of the receptors. So once it is formed, tyrosine is activated. Okay, once the tyrosine is activated, it will now cause the ATP to become ADP, which causes your phosphorylation of the receptors. It causes phosphorylation of your receptors. Once its tyrosine picks up phosphate group, different inactive intracellular proteins come up and attach to the phosphate tyrosine. There, the conversion of your ATP to your ADP, leading now to cascade of activation, then they will produce a cellular response. And this was what is happening to your enzyme link receptor. Now, in G protein, okay, in G protein, first, you can see that class, the ligand, okay, as you can see, the ligand first bind to the receptor. This ligand class, as you can see, it will bind to the receptor. If this ligand bind to the receptor, it will cause a structural changes. It will cause structural changes into the receptor. There, extra structural changes into the receptor, and then turns changes and shape of the G protein, which reduces class GTP to GDP. Okay? Leading to the separation of the alphas, you can see alpha subunit from beta and gamma unit. You see that, class? Okay? I'll repeat. So first, the ligand will bind here to the receptor, causing structural changes into the receptor, and then changes the shape of the G protein. which reduces your GTP to GDP, leading now to the separation of your alpha. As you can see, the alpha separated. Okay, alpha unit separated from beta and gamma unit, and it turns, which turns activates other enzymes and your G proteins. Okay, and your G proteins. Now, how about for the fourth type and the last type? That is your intracellular receptor. This is very simple. In intracellular receptor class, okay, this particular ligand that pass through the cell membrane enters, okay, to the cell and into the nucleus, as you can see, crossing the nuclear membrane. So after entering the nucleus, the ligand now will bind to the intracellular receptor there. Perfect. Okay? If that particular ligand binds into the intracellular receptor, this now can alter the transcription of DNA within the cell. Okay? So those are the four types of your receptor.